Hello. Good evening, everyone. Today we are going to discuss about a new topic like it is extension of mutation breeding. I thought to discuss varietal release, but anyhow I started mutation breeding, but I will end by advanced technologies today and tomorrow. Okay. Before starting, we'll start the class without any delay. Without any delay. Yeah. Till today we have discussed various breeding methods in self and cross-pollinated crops, and we have discussed transgenic breeding. We have discussed molecular breeding, and we have discussed hybrid breeding. Okay, but all these technologies except transgenic breeding and molecular breeding, and most of the technologies are developed like in 19 not in 19 not in 19 not 10 19 after 19 not 10, like after the rediscovery of the Mendel. Then when you think like nowadays breeding, like when you compare the breeding objectives of then and now, there will be a great difference. Nowadays we are breeding for enhancing the productivity under changing climate and with a less resource. But earlier time, we used to breed for enhancing the productivity. Like earlier time, like well, take the example of Green Revolution time. At that time, Indian cultivars are not that much like higher yielding, not productive, and we are suffering from food food security was not there. Our poverty, hungriness is one of the biggest problem. The breeder objective generally it was to increase the yield to meet the immediate need of the country and to meet the food security. But now we have self-sufficient and we are looking forward enhancing the productivity and in the climate change. Like earlier, it was not that much problem. We have like with various resources. Like at that time in 1947, what is the like population of India? Can anybody guess? What is the population of India in 1947? Earlier in 1900, no one is there. Satya?
I think 50 crore something.
Yeah, it's around 30 crore, 50 crore. Okay. But now today's population is 150 million. I think it's about to 150 million. See how much 30 million to 150 million it increased. But the land we have, like Bangla, like separated from us, Pakistan was separated from us. We are now in 2015 also we transferred some enclaves to Bangladesh. This land is continuously reducing, but population is expanding in an exponential way. Like in every paper we will write the first sentence, like increasing population, changing climate, decreasing resource, same way. It's all real. Okay? Because to changing the climate, land is minimum that we have to increase the productivity. For example, you take the rice. Rice in who is the highest like who having the highest production of rice in globe? Who is
China, sir.
leading? China. At China, within a 30 million hectare area, I think around 20 to 30 million hectare area, it producing the more than us. But even though we having 40 to 15 million hectare area, we can't produce that much. Even though we have larger production, but when it comes to productivity, it's very low. But in future, but population size of India is growing exponentially. We can't offer that much of land for production of rice in future. That's why now the breeding is challenging. We are targeting and higher productivity with combating with this urbanization, climate change. Okay, then at that time the breeding, like earlier, they were more focused on like responsive, like NPK responsive. During Green Revolution time, we normally bred the varieties for should grow in higher environments, so that immediately the production will increase, so that we can feed the population. But nowadays, we are more talking about efficiency, like use efficiency, like water use efficiency, nutrient use, fertilizer use efficiency. Why? Because we had to grow in a sustainable way. Now we are self-sustained in the food production. But now we are facing this climate change. Means we have to adapt the some methods which are sustainable in terms of environment. That's why we are more talking about efficient breeding, like water is efficient breeding, nutrient use efficient breeding, sustainable breeding. That's why our focus is shift from nutrient responsive to the nutrient use efficient and the resistant breeding. Earlier, we used to breed for a single resistance and dual resistance and with minor pest and diseases. Like rust was the major, like some bacterial blight was the major interest. But now we are having multiple discs. See the pink bulbworm in cotton. Nowhere nobody thought that it will become that much devastating pest. That's why new pests are rising due to climate change, due to change in the environment, weather pattern. We had to continuously breed for the dynamic change in these pests and diseases. That is the one thing. Then another, abiotic stress tolerance. Like earlier, there is because of this environment stability, we don't have that much pressure like heat stress, drought stress. But nowadays, we have to breed for multiple abiotic stresses. Okay, and without compromising the yield. And earlier days, we are breeding for like no emphasis was given for nutrition breeding. You check the papers like before 1900, like 1949, 50, maybe color, maybe it will be the preference. But no much varieties released for high protein content, no much varieties released for high zinc content. But today you take like most of the papers you read, most of the newspaper, we are more focused towards like enhancing the grain nutrition, like how we come across from food security to the nutritional security. Breeding objective has changed. Okay, from like we are more focusing on the improving the nutrition in the crop. Okay, then and now we are talking about we have to meet every country in the globe has to meet several sustainable goals by 2030. Like we had to reduce the poverty, we had to be sustainable with the environment, we had to become a clean energy. And at what year India committed to become a zero emission state, like zero emission country? At what year?
2047, I think so.
Yeah, no, 2070, I think. Once you check, 70 or 47. 47 is Amrit Kal, but 2070 is ultimately we fixed that we will become a zero emission state. Okay. Then we had to, if a zero emission means what is we had to have a low, we had to grow crop with a lower carbon, like a low carbon footprint. But how we are breeding? Like for rice, you take how much methane it will produce and how much nitrogen we have to keep. Like urea was the one of the fertilizer we have to apply and how much soil residue, like fertilizer residue it will leave and how much methane it will reduce. Actually, in European Union, actually have restricted the import of our basmati basmati and both non-basmati rice because it having larger carbon footprint, like it emissions more with like they estimated like 27% of methane emission is due to rice production. But greatly the Indian scientists like now present DG has proved that it is not that much contributor for the greenhouse gas emission. Like he has actually had that much contribution for the Indian farmers. Like present AG, who is present AG?
Himanshu Pathak.
Himanshu Pathak. Okay. Then nowadays we have breed for that. Then earlier we have focused on like a yield only, but now we are focusing on product profile. Like for example, we are already become self-sufficient. Whatever we are producing, meeting along with the meeting domestic needs, we are more focusing on export. To export means we had to meet all the standards of the different countries. Means we had to talk about product profile. For example, you take last year, like, I think in this year, last year, European Union again banned all the Indian spices because most of the spices contain the preservatives which are very carcinogenic in nature, like carcinogenic in nature. But we have to meet like how we can reduce the preservatives, how we can test them, and how we can develop the like lines which are more perishable in nature to avoid adding the preservatives. So many aspects we can breed. The product profile is becoming very important. Those crop we are mainly targeting for the export. These are all the like now to breed all these breeding methods. If you use the earlier method like pedigree breeding, bulk breeding, or your conventional method, like it's like for you take the example, if we are in a warfare, like in war, some countries soldiers having only a pistol, and some countries soldiers having a pistol with AK-47 grenades and everything. Those who will win means normally the more the soldier which having the more weapons and more resources, then they only win. In the same way, along with the earlier breeding methods, we should have an uh, extra toolbox or extra weaponaries to combat all these limitations of present world and to improve all present breeding objectives. That's why we need a good method, like we need additional methods to combat all these limitations. That's why we are more focusing about nowadays. Earlier we are only crossbreeding, transgenic breeding came, molecular breeding came, mutation breeding came. Now we are landing into genome editing. Now we are talking, everyone is talking about genome editing, like how we can breed through the novel technologies so we can meet all the immediate present breeding objectives with more efficiently, more sustainably. That's what like genome editing. Yesterday we have talked about mutation breeding. Like generally it's a random mutagenesis. If it's a random mutagenesis, definitely it will take a lot of year to isolate and select we can do, but isolate and bring them into homogeneous condition and releasing it takes at least 8 to 10 years. Like in crossbreeding also, what we will do, we are crossing and we have every breeding method you take at least it need 8 to 10 years. Then transgenic breeding, we have talked about transgenic breeding. Like it also at least take 8 to 12 years because you have to ready the first you have to identify the target gene, then you have to identify the source transgenic gene, then you make the construct, then you have to integrate, then you have to develop, then you have to go for all your uh, safety protocols, biosafety protocols, then it will be released again. It having a very stringent rules. Still only one crop has released in India in terms of transgenic varieties, which is that it's a cotton in 2003 or 2002, I think it is released, but no other varieties are released, even though it having a great potential. Because of these biosafety and environmental safety conditions, it is not deregulated. Then what will do? We can't, even the transgenics have better approach than mutation breeding and crossbreeding, but we can't, no varieties are going to release. Everyone filed the case in the Supreme Court to not release these varieties. Okay, then what we can do, what breeder can, we can't use transgenic breeding only for the basic basic studies we can use transgenic breeding, but we can't release as a varieties. Then what else we can do? Then scientists came up with another approach like genome editing. Like we can breed by using the novel technique called genome editing. It is like it is a mutation breeding only, but earlier mutation breeding is random. But here, in this mutation breeding is more targeted. Means we are not altering where we know that where we have to make the mutation and where we need to select. And it is just take 2 to 5 years, and it reduces the year like from 8 to 10 years to the 2 to 5 years. And also it is precise in nature, and also there is no required of backcrossing. It's a very not laborious, and it is transgene free. It is not like it doesn't contain any transgene. That's like we can all thus all environmental safeties can be it is not overcome any environmental safety regulation. That's why nowadays most of the scientists are focused breeding varieties to meet immediate objectives of present day with the novel technique called as genome editing. Yesterday we have talked about mutation breeding. It's random in nature, but today what we are going to discuss is more targeted, more precise, and more efficient. It's about time and labor. Okay, then what is this genome editing technology? There are different technologies. That's why I wrote. There are nowadays not only a CRISPR, there are several technologies. But there, normally we think genome editing is only one technology called as CRISPR. But it's not like that. There are several technologies of genome editing. So I wrote genome editing technologies. Then what is genome editing? It means that it's a targeted intervention at genomic sequence. I'm just telling genome editing. Not I'm telling genome editing breeding. Genome editing means it is just targeted intervention at genomic sequence. How we can intervene at targeted site? Either by inserting, either by deleting, either be manipulating by specific trait by inserting, deleting, or manipulating some genome, like a genome part. Either may involve introduction of foreign genetic material or it may not involve. Like there are several modification of gene editing. Either it can introduce a transgene or it could be a transgene free also. It depends on the need. Understood what is gene editing or genome editing? It is a targeted intervention at the genome sequence by either making insertion or deletion manipulation of the genome to alter specific trait. Either it involve introduction of gene or it may not have introduction of transgene. Understood these points? It's very important points. Okay. Any doubts apart from this slide?
No, sir.
No. Then how we can proceed? Like what are the steps we follow? First, first you have to identify the gene. Then we can construct a target site first. Then based on that we can design the target. This one we will discuss. Then we will vector construction. Then you validate that one. Then you introduce into the plant. Then you generate the plant regeneration. Then you screen it and then you make phenotyping to study either your target trait is altered or not. Then you check for the transgene is there or not. Then you go for release. First, you have to do, you have to identify the gene. Then you can construct a design for that, but you can design particular mRNA, sorry, siRNA for that particular target sequence. Then we can design a vector for that. We can multiply and validate that. Then we can introduce into our plant cell. Then we can regenerate the plant in a tissue culture. Then we can screen them. It is like Sanger sequencing. Either you can screen with PCR, either you can screen with Sanger sequencing. Then you validate it. Then you release, like you release for trials, field trials. Then after that, it will go for normal system of release that we will discuss tomorrow. Okay, this is what the broad picture of genome editing. Okay, then what are the steps? For example, I hope like we will discuss first what is how to what are different technologies available to edit the genome. Later we'll come to this. Okay. Then what are the different genome editing technologies? There are earlier not only CRISPR. Nowadays we are talking about CRISPR. Before that CRISPR, there are several technologies like meganucleases, zinc finger nucleases, and TALEN. These were the three techniques were available, and these techniques came after 2009. Like in 2009, the actual discovery of CRISPR-Cas9 has taken place. The who was the scientist who was actually major contributor of developing CRISPR-Cas9 technology?
Jennifer Doudna.
Yeah, actually they combined both CRISPR and Cas9 to edit the gene. But earlier only CRISPR was there, Cas9 was there. But the involvement of these together with editing the genome has came after discovery by Jennifer Doudna and another scientist called as Jennifer Doudna and Emmanuel Carpentier. Okay, then what we all the and what is the difference between these three techniques and this one technique? How why only this genome editing technology become this much famous? Anybody know? You can tell the answer. Why these technologies are not that much famous? Why this technology is become boom actually? Why?
Sir, in CRISPR, there is guide RNA like that. It will identify precise sequence in the DNA.
Yeah, that one. It is more precise and less off targets that we can in terms of it having like according to your point, the one word for that point is of its minimum off targets. Then any other? Now the important reason is, and these are techniques, you have to design the proteins to target the particular genome. But Here you are designing an RNA to target the particular gene. Designing RNA is very easy. It's just a nucleotide. You just because it's stable in nature and making nucleotide sequence is very easy. But designing the protein for every sequence you need to design separate proteins. Okay, like had to had to make design of this protein which able to bind with the target sequence. Designing proteins is very easier, or designing this nucleotide sequence is very easy. Which one will be the easy? That is the like designing mRNA sequence is more like easier than the designing the proteins to target the particular gene. That's why CRISPR-Cas9 has become more popular than the other technologies. Then, like its off targets were very less because what is off targets? Because you have designed this particular uh, either it may be meganuclease, either maybe zinc finger nuclease, either maybe TALEN, either it may be CRISPR-Cas9, like your CRISPR, to target this particular site. But instead of this site, it may also target for the another site. That is called as off targets. That off target like off targets will be very less in case of CRISPR-Cas9. And that's why it is more precise and less off targets. And designing RNA is very easy than the designing the protein. That is one of the greatest advantage of this CRISPR-Cas9 based technologies than the other technologies like meganuclease, zinc finger nucleases, and TALEN. Okay. Then, like FOC1 enzyme was actually the nuclease in this method. Here, the Cas9 was the enzyme which actually act as a nuclease. That one point to remember. Like this here, FOC1 enzyme. Here, Cas9 enzyme was the nuclease. Then, zinc finger was actually bind with the target. Then, it will allow FOC1 enzyme to break the sequence to introduce double-stranded break. But here, the CRISPR will attach, the CRISPR RNA will come on target. Then, again, it come with the Cas9 enzyme. Then, Cas9 will act as a nucleus and it will introduce double-stranded break. Okay. You understood what are the different tech. I'm not going in detail about what is meganuclease, who discovered it, and what is the mechanism of genome editing of meganuclease. It is out of like it's not it has to be studied by biotechnologists. For a breeder, you just remember along with the CRISPR-Cas9, there are other technologies like meganuclease, zinc finger nuclease, TALEN was there, and how we can able to introduce the double-stranded break and what happens after double-stranded break. This is our target of interest. Okay, how it can modify the genome, that is our target of interest, because here change will happen, then definitely there will be change in the phenotype that we want. But what tools we are using, that is not that much necessary for the breeder. But again, this year the question was asked about base editing, prime editing. We will discuss tomorrow's class. Then, understood what is the different technologies available before CRISPR-Cas9 was meganuclease, zinc finger, and TALEN. Understood?
Yes, sir.
Yeah. Then it introduces the double strand. But okay, it creates something not normal to the cell. Like it's creating the condition which is not normal to the cell. Cell has to repair. What is the different repair mechanism available now? Like what is the different DNA repair mechanisms? At least you should tell subheadings.
Base excision repair.
What? Yeah. BR.
Nucleated. Nucleated excision is there.
Okay.
Recombinant repairs.
Another one.
Direct repair.
Exactly. Means what happens in the repair? What is repair? There are n number of repair methods. There, what happens?
The damaged DNA is excised and new DNA is synthesized.
See, whenever defining, you should be very precise. See, always it's not the case where new DNA was inserted. Sometimes what will happen, it will just modify the present DNA, or it may take the homologous sequence, then it will add. Okay, it is inbuilt mechanism. It will not add any other new DNA to it. It will happen. I'm not telling in recombination repair it will happen. No, HDR it will happen. But to take NHEJ, we'll discuss where new DNA is not introduced at all. Okay, you just modify some bases, then it will combine. Then that's why your mutations will come. It is one of the source of mutations. Okay, please read what is the DNA repair chapter. It's very important for exam point of view. Those who are preparing CSR, it is very important. Then what is like when the day, as you said, there will be repair mechanisms normally. When you are introducing double standard break, there are majorly two types of repair mechanism along with that. One is non-homologous end joining and homology directed repair. Non-homologous end joining means here for repair there is no any template. Okay, repair is like break was happen. Somehow it modify and it will attach this one and it will overcome this damage. Either it may add some deletion, either it delete some sequence, sorry, delete some bases, or it adds some bases, but somehow it will manage. Like it will make stitches, then it will create normal DNA. That's why it is more error prone because it's a non-guided repair of your double stranded break. Remember, NH non-homologous end join in the name itself is means it doesn't depend on homologous sequence, like it acts as a template. It's a non-homologous and you are joining the ends of the DNA. Okay, then you are repairing this break by this method is called as non-homologous end joining. Then it may add some insertion or it may delete some it may add some bases or it may delete some bases. That is called as entirely it is called as non-homologous end joining repair. That may be that definitely may be error error in nature. It will create errors in the cell. That's why your mutation will arise. That we can use it for trait improvement. If it is a favorable mutation, that use it for trait improvement. Understood? Non-homologous end joining. I think many of you read already, but take it as a revision. Then, then the another method is like homology directed repair. Understood what is NHEJ? Hello.
Yes, sir.
Yes, sir.
Yeah. Then another method is called as homology directed repair. Means we are giving a template to repair this double-stranded break. Here, in earlier case, it was not like this. You are giving a method, like we are not providing any such template to repair. You are allowing itself to repair. Like cell itself make repair. Here, a cell uses some template to repair your DNA, like double-stranded break. That's why it is because what changes you want to make in this particular DNA, you can guide them. Okay, you can guide the cell. Okay, you have to you have to insert or you have to mutate the DNA in such a way that you want because you are provider. Because along with the construct of CRISPR-Cas9, you are providing a template also so that your repair can be takes place according to the provided template. That's why it is not that much error. Okay. Even this will not create an error during the evolution. Why cell is why the NHEJ is more predominant in cell? NHEJ is more predominant than the HDR. Why? Why is it so? Anybody know the answer? Can tell. Can anyone tell the answer? Hello.
Maybe the template strand is not available.
No, actually NHEJ occurs throughout the cell cycle, but HDR will occurs only during S phase of the cell cycle. Okay, that you should remember. It's an H like in a cell, the HDR will happens only during S phase of the life cycle, cell cycle, but your NHEJ occurs throughout the cell cycle. That one point you can remember. Okay, why if anyone asks you that why HDR is less frequently occur in the cell than the NHEJ, it because the HDR will happens only during S phase of the cell cycle because it's needed template like while DNA replication only. It is like producing some template like structures. Based on that, it will guide like it will overcome the mistake. Okay, that's why the HDR will happens in S phase of the life cycle cell cycle, but NHEJ occurs throughout the cell cycle. That's why NHEJ is more predominant in cell repair. Okay. Then understood what is gene edit, like what are the editing technologies are available, which one is famous, why it is famous, and after the break, how cell can overcome this repair by two methods called as NHEJ and HDR, and why HDR is more like NHEJ is more occurrence occurrence of frequency in the cell. And you understood all these points.
Yes, sir.
Sorry. Yeah. We will talk about CRISPR. Okay. You know that? Okay. Now, actually, in 2022, one question was asked, which of the following is not a gene editing technique? CRISPR-Cas9, zinc finger nuclease,
Gene
TALEN,
gun.
and gene gun.
Gene
Yeah,
guns.
gene. See, simple questions. Normally, we read lot of things, but the questions should be very, very easy that we cannot remember. Like, we'll confuse. And scientists were Jennifer Doudna and Emmanuel Carpenter, and they awarded Nobel Prize in, I think, in medicine in 2020, I think. Correct? 2021.
20 only.
Just check it. Then what is CRISPR? So we studied about this is the one of the gene editing technology. What is then CRISPR? Like in 2023, the question asked directly, like what is CRISPR? It is cluster regularly interspaced short palindromic repeat. You should remember each and every word. See, in the options they have given clustered regularly interspaced short palindromic repeats. Clustered regularly, see regularly. Normally while reading also I just read regularly also. Then I forgotten this two or miss. I was not that much conscious about reading this. That's why in exam consciousness is very very important. Even though you know everything, you may do mistakes if you don't have any authenticity if you're not attentive in the exam, these mistakes will happen. This clustered regulatory interspaced short palindromic repeats associated 9, it is not there because it's not a regulated. Okay, then clustered regularly interspersed, it is interspaced, this is also wrong. Then clustered regulatory interspersed. It is again wrong. It is not regulatory. Clustered regularly interspaced short palindromic repeats. Okay. Now understood?
So,
Yeah. Fourth one is the option. Anybody did mistakes here. Okay. Like you are losing 5 marks. If you are attending negative and mistake means you have 5 marks. Then what is this actually? Why it is clustered, regularly interspaced? Means it is clustered in the genome. Cluster means what? What is the meaning of cluster?
Groups.
Yeah, it's a group. Means it's a one group. And it's occurring regularly in the genome. How it occurring? See, it's a group. You take group and it is regular in the genome and it is interspaced. Means this group having something in between and there. Means interspaced, having something between them, it is there. And something and it is clustered and regularly interspaced means this interspace is regular in nature between this repeats like short, and it is a short palindromic repeat. I think you know what is palindrome. This is what CRISPR, clustered regularly interspaced short palindrome repeats. This is a unique, actually this was earlier identified and it's one of the unique nature and is a part of immune system of the bacteria. By using this particular sequences, it invades the virus and it will kill the virus DNA. How it will act, we'll see. Then it's like it called as a primitive warfare between the bacteria and the virus. For example, if you take the bacteria, whenever the bacteria phase, what is bacteria phase?
The viruses which attacks bacteria.
Yeah, virus that attacks bacteria. Generally, virus feeds on bacteria. That also. Well, then this particular bacteria phase, whenever it infects the bacteria, it will have some genome. This is, for example, it is a genome. And whenever it infects the bacteria, after infection, the genome of this virus will definitely integrate into the genome of the bacteria, like DNA of the bacteria. See this green color, whenever it infects, it will come and introduced here as a spacer. Then if any this next one is, which is this color like blue color, pale blue color. If it introduce any DNA with pale blue bacteria, like DNA having pale blue color, bacteria is introduced. Here it is introduced again spacer, and this particular DNA part introduced to bacteria DNA as a spacer. If any green color bacteria phase will come, this particular part of the DNA will introduce as a spacer. Again, if any brown color like yellow color will come, again it will introduce as a spacer. This one are from these are from the bacterial gene, like bacteriophage gene, viral gene. Whenever the virus infects the bacteria, the viral genome, some part of the viral genome will introduce as a spacer in the bacteria in a very regular order. And this cluster and regularly interspaced sequence is called as CRISPR, along with Cas operon and tracer RNA. This entire locus is responsible for immunity. It creates the immunity for the virus. How it will create? For example, now it having the green color, blue color, light green color, and light yellow color. Whenever the DNA with the green color will come again, suddenly what will happen in the host? This particular loci will transcribe, entire locus will transcribe, and it will again degrade. Then there will be synthesis of CRISPR RNA biogenesis. Then it will accumulate, like for example, again green color will come, green color bacteriophage will come. The green color microRNA, like chromosomal RNA, sorry, CRISPR RNA, will associate it with the Cas9 protein. Then it will come and bind, sorry, first it will recognize, then Cas9 will come and it will degrade your viral DNA. Okay. If you're not understood also, please watch the video. How like one like there from Nobel some what is that Nobel, they have those who will award the Nobel, they have a YouTube channel. like I think you also watch like Nature YouTube channel. Nature YouTube channel was there. Nature Publications. Their in detail animation was very good actually. If you watch this video, if you not understood like how actually the CRISPR array will act as a immune system for the virus. At least understood some points. What is there is a tracer RNA, Cas9 local which produces the Cas9 engine, and CRISPR RNA. Okay, it produces CRISPR RNA. CRISPR RNA and tracer RNA together will produce CRISPR RNA. Earlier it is called as pre-CRISPR. After it will combine, it will produce CRISPR RNA. Then it will target the host. Then it associated with the Cas9. Then entire it is called as CRISPR complex. Understood what is the how it will what is the mechanism of CRISPR Cas9 as the immune system for the bacteria? Any doubts? Even though I'm not clear, watch any animated video, it will be very, very clear. It is simple. Okay. First, initial time, whenever the bacteriophage is introduced to the genome, the particular genome of the virus will introduced as a spacer. But if again the bacteriophage come and infect the bacteria, it will produces the CRISPR RNA in relation to like in correspondence to the genome of this bacteriophage, and it will degrade the bacteriophage DNA. Then it will protect it from this particular phage. This is a simple mechanism. Understood? Any doubts regarding this? I hope no doubts. Okay.
No, sir.
Okay. Then, what are the system like? What is CRISPR system contains? We'll look into in detail. First, it having a single guide RNA. Earlier, it was one part is tracer RNA, another part is your CRISPR RNA. Sorry, pre-CRISPR RNA. Then, when it is become like after this one, then it will become single guide RNA. Then, after it associates with Cas9 protein, and it recognizes the sequence which are based on which is adjacent based on the PAM sequence, protospacer adjacent motif. If it is a PAM, how the single guide RNA, like first there will be tracer RNA and pre-CRISPR RNA, then both will normally in bacterial system we have two types, tracer RNA and pre-CRISPR RNA, then it will become trace CRISPR RNA. But in artificial system we having only one RNA called as single guide RNA, means it having both your target specific sequence and tracer RNA. By removing unnecessary sequence, we are making single guide RNA, because we should have narrow, like whenever you having a larger this one, then it is very difficult to bring that to into construct. It is very difficult. That's why we had to make smaller. We are removing unnecessary part. Then it will be a only containing target specific sequence and tracer RNA backbone. Then it is completely called as single guide RNA. Then it actually comes associated with the Cas9. Then wherever it, how it will target the genome based on the PAM sequence, like protospacer adjacent motif here also. Because every spacer having PAM, after only recognition the PAM, then it will target the genome. Your CRISPR single guide RNA will target the genome based on this PAM presence of the PAM sequence. Okay, you should many times ask in the exam. PAM sequence is very, very necessary. Even if you are designing single guide RNA, it is based on the PAM sequence. Okay, then it is target your this like a genome, like your favorite gene. Then, like after targeting, it will introduces the double stranded break. Then cell will repair either through any of the mechanism, either non-homologous end joining, either HDR. This is about the components of CRISPR RNA. It having single guide RNA, Cas9 protein, after association, will target the gene, then it induces the break. Understood this mechanism? See, lot of
Yes,
like
sir.
we have done 6 months course for genome editing, but I am making short in 1 hour video, like as which is required for the exam that I am teaching. But in PhD, we have separate course for genome editing only. You can read in detail, like what are the, see, to removing extra portion, there is a lot of things. Like there will be how we can remove that extra sequence. What is that sequence? Okay, how to design this one? Which tools we are using to design the single guide RNA. Like how, what are the different now? NGG was normally used PAM sequence. Along with NGG, there are several PAM sequences are there. How to do that? There are a lot of things were there. Again, in a Cas9 protein, how actually when Cas9 and single guide RNA was combining, what modification will happen in the Cas9 protein? Okay, what are the molecular mechanisms will happen? Then what is the structure of this Cas9 protein and which domain it will have? It is having two domains, RBC domain and HNS domain. And if you modify HNS domain, what will happen? If you modified RBC domain, what will happen? A lot of things were there. I will share on paper a CRISPR toolbox on paper and genome editing for feature breeding. I will share two papers. If you read that papers, you will get like Cas9 structure. It having, you know, this is a protein having multiple domains. It having NUC lobe, REC lobe, and NUC lobe. Then it having helical domain, then HNS domain, REVC domain. See, why it is very important? Means REVC domain, HNS domain. Based on this, we can modify, and there are other tools for development, like base editing and dead Cas9. If you modify, if you mutate both the domains, it will become dead Cas9. It will not introduce the break, but although you can use it for many other purposes. And you can mutate only one of this domain, it will become only single strand cutter. Then we can use it for any other, like in base editing we will use this one. We will mutate one of the domain, then we will use it for the editing. Either you can mutate both of this domain, either you can use it for like gene tagging, either you can like you can methylation suppression. There are a lot of things we can, lot of things we can do. Okay, that's why you just remember what the two domains, RUVC domain, HNS domain, and where your PAM sequence was there, and how actually this components, if you remember, that is more than enough. Then, like how you will do, like what are the mechanism, like you have, you know, what are the components you needed, then how you will introduce into the plants, we will discuss. Then you have to make the construct first. One is your booster and DNA. Like a very n number of method is just one of the method normally used. Then it's like agrobacterium mediated transformation. First, you construct the DNA which having CRISPR genes, Cas9 genes, and your all your Vir genes, all these things. Then you go for agrobacterium mediated transformation. Then you just do selection. Then is develop your genomic in, if you don't have any agglomerated transformation methods like monocuts, it is not that much responsive. You just by using like biotic transformations, like ballistic transformation or gene gun method, other tungsten method, whatever it may be, you can use other like transformation independent technologies, also floral dip, whatever it may be. But you have to introduce this construct into the plant. And in detail we have discussed what the different technologies were there, what are the advantages and disadvantages in detail. I'm not going again. Then you just select the plants and you can release. Then Hawaii, you are introducing the constructs, you are introducing your recombinant DNA into the plants. But while we regarding genome editing as transgene free. Why? Why is it so? Here we are doing all whatever this transgenic breeding we are doing, same thing we are doing here. Like we are introducing recombinant DNA, we are selecting, we are having marker genes, okay, selectable marker genes. For if you want to select means you have to having selectable marker genes, okay, scoreable marker gene, selectable marker gene, everything protocol is same. But although why we regarding genome editing plants are transgenic free?
To overcome the regulatory restrictions.
Yeah, we will discuss. Okay, that one point you remember. I have slide. We will discuss. First, I will discuss in detail about how we can. What is the construct we have to make for the your genome editing. Now, first you have to choose the target site. First, you have to choose the target site. Then you have to design guide RNA for that. There are many tools like ChopChop, or you can use any other tool. Like ChopChop is mostly used tool. ChopChop. Either you can synthesize guide RNA. Then after that, there is a one technique called as codon optimization. Why like codon bias? Anybody heard about the term codon bias?
No, sir.
You not read medicine.
Yes,
Genetics.
sir. Sir,
Yes,
like
sir.
one coding for more than one type of amino acid.
See, that is what is that nature it is called as?
Ambiguous.
Degenerative.
Yeah, you forgetting all the basics. It's a degenerate nature. But why we need to do what is codon bias?
Sir, when specific organism
Yeah,
prefer
you told
specific
right answer.
codon
You
to
are on
insert
the
that
way.
amino.
Can you repeat? The degenerate nature is good. It is related to codon bias. But how? You should come up with proper definition. Well, not proper definition. At least you are in the middle of the point. You have not reached the destination.
Sir, codon bias is when a particular species or organism prefer a specific codon to insert that amino acid.
Exactly. Exactly. Okay. For example, because we are having see how except AUG and another sequence, most of the codons were degeneracy nature. Degeneracy nothing but one codon may specify like different amino acids. Sorry, like one amino acid be specified by different codons. Okay. In for example, for synthesizing highest codon available for which amino acid?
Is a serine, sir, or I forgot, sir.
Take
Serine.
anything. Okay. Serine. Then means you are for example maximum 6 codon for serine you think. Then in one species one codon used for particular amino acid. In another species one codon is for that amino acid this one. In another species one codon is preferred over the even though there is 6 codon codes for single amino acid, but although in some species they prefer only, for example, you take GGU code for one amino acid like GGU, GGG, GGC, GGA, all these codes for phenylalanine. So I will write, it will be very clear. GGG, GGC, GGA, GGU. All these codes for one amino acid. You just think. This is degeneracy nature. But in some organism one, this codon is preferred. In organism 2, this codon is preferred. In organism 3, this codon is preferred. In organism 4, this codon is preferred. This is called as codon bias. Means some species having only preferable preference to the one type of codon producing the amino acid. It does not prefer all these 3 other codons. This is called as codon bias. Means the Cas9 is from the bacterial origin, but you are putting this Cas9 enzyme into the plant origin, like plant cell. Means in Cas9, when it's Cas9, this particular this one, for example, some E. coli or any other organism which having, like you should remember Cas9 from which organism, like Pseudomonas. Uh, I have forgotten. Please remember organism also. From where Cas9, which bacteria Cas9 came, Cas12 from came from which organism? It's very very important. Like I forgotten. Okay. Please remember, read actually. You and I also read. I will also read. Okay. Then, what will happen in some organism? Cas9, like synthesizing a Cas9, like preferred by one codon, like GUG. But when you are bringing this Cas9 producing gene in the plant cell, then definitely it will prefer other codon to synthesize this Cas9 protein. Means there will be difference in this codon, this one. Then definitely you may not get a proper Cas9 with the same effectiveness like in the bacteria in the plant because of this codon bias. That's why before introducing the genes of Cas9 into the plant, you should optimize so that the efficacy of this particular enzyme will not disturb. That's why you should do codon optimization of any enzyme before introducing from one species to other species. This is called as to overcome the effect of codon bias, you have to optimize the codon, which codon is the particular species using, and try to modify accordingly. Then you introduce. Understood what is codon optimization and codon bias?
Okay, sir.
Then after codon optimization, then after your single guide RNA synthesis, then introduce like both of genes into a construct. Like your construct look like this. It having a Cas9 producing gene. It also having your gene producing like your sorry single guide RNA producing genes. Along with that, you should have a NLS. Why we need what is NLS?
Nuclear localizing signals to
Exactly. It is called
put
as
them into.
nuclear localization signals. Means this nuclear localization signals, what will make, what is the function of this NLS? And why it is only like, why it is associated with Cas9? See, in a cell, if you are introducing this construct, Cas9 gene will produce Cas9 protein, and this will produce RNA. How this will come together in a cell? Who is guiding to Cas9 or who is Cas9 to go and bind with your single guide RNA? It may go and bind to any other sequence. This nuclear localized signal means it having some path like it's act like a signature protein. Last time their question asked target signature sequences. Like it's called as protein targeting. After transcription, the particular after translation, how these proteins are going for particular organelles. It is called as protein targeting. In the same way, it having some signature sequence here, nuclear localized signals. It will bring this Cas9. It will make sure that Cas9 will go and bind with the guide RNA. Otherwise, it will be aimless. Where it will go and where it will bind. That's why we should have NLS sequence along with the Cas9. Then, and you should make sure that again this kinamycin resistant for a selectable marker and HPT for a selectable marker, and you should have promoter sequence, 35 promoter. See, every gene construct should have, because it's an expression vector. There are two different constructs, two vectors. One is cloning vector and another in expression vector. If you want to express the particular vector, because you are expressing, you want to produce RNA and the Cas9. That's why it is called as expression vector. Means if expression vector should contain your promoter genes. Means if promoter will transcribe all these genes, Cas9 genes and this one, and you should also have terminator. Here you have terminator. Then you should have tDNA, and it should be all things should be included within the which region of the type plasmid.
tDNA region.
Yeah, tDNA region means it should be between the left border and right border. Okay. Then you introduce this one like this construct. Actually, it is in the plant cell. Then it will express. Then it will produce single guide RNA and it will produce Cas9 and it will combine together and it will based on the PAM sequence recognition. It will go and bind. Then it will introduce the double strand. This is how that is. Then this is like what is normal transgenic protocol, like how to regenerate plant, how to then how to screen that mutation. Then everything is same. Understood? Then you are making everything like you are introducing marker genes, you are introducing promoter genes, external promoters, you are introducing external terminator genes. Then why still it is a non-transgenic? Why? Because after that, what will happen? We will cross with normal individuals. This particular mutant, transient, like created by the genome editing, it cross with a normal mutation, like normal lines. Then we will allow it for segregation. Then we can select the plant. See, these are the marker genes. Like what is the foreign gene, but with mutation. You may get possible segregant was either like either there are two chances. First, mutation and you're all transgene in the single chromosome like this. Sorry, like this situation, or your mutation in one part of the chromosome and another part of the chromosome, your marker genes were there, all transgenic genes were there. These possibilities. Either in some gametes, you may have having only only mutation without marker gene, and in some gametes which having only like no mutation, no transgene, nothing. It's normal gamete. And here we having only gamete having only marker gene. All four types of gametes is possible in this case, and all four types of gametes possible in this case. Either they are in the same gene mutant and marker gene, then like after that what will happen? When you are crossing with a normal individual, it will segregate. And you have to isolate the particular lines in such a way that you should only having mutation. This red star is only mutation. But all your marker genes will be, you are not selected. It is simple segregation method. But to do this, we need at least, like in rice, in IRR, they have used nearly 3,000, sorry, not 300 to 400 population to isolate this particular one or two individual which having only mutation but not having all these marker genes or transgenes. That's why the varieties which are released, like varieties which are developed through this genome editing technology is non-transgenic in nature. It's not mean that it is always non-transgenic. If you are targetingly introducing particular genome sequence, this mutation may be a single base in SDN1, sorry, if it is from NHEJ, it is maybe introducing, like NHEJ means it's not having any external template. Either it may introduce a small template, it is also non-transgenic. But it may also introduce a large chunk of the GNA into the background of the genome, it will become transgenic. But normally, why it is not transgenic? If it is think that you are changing only few nucleotides, why? Because after segregating, while segregating generation of large population, you are isolating the plant which having only the mutation but not the target gene. Like sorry, transgenes. How we can do that? How we can screen that? There are several techniques. Either you can use normal PCR, either you can use uh, uh, okay, sorry, I forget to tell. What are the detection method for the transgenic? There are several I discussed earlier for transgene detection methodologies. Can anybody tell if you know what are the different technologies available for transgenic detection? No one is there. Okay, let it be. Yeah, there are several techniques. Either you can use RT-PCR method, either you can PCR method, either you can use a Southern blotting technique, either you can use any advanced techniques for like for detection of chip method. Chip methods are there to detect the transgenics. Okay, based on this any detection method, you can check either presence or absence of transgene in this population. Then wherever you are getting no transgene, only desired mutation, then you release as a variety. This is what the different transgene. Okay, like why it is the genome edited crop varieties are non-transgenic in nature. Okay, I think understood all the topics like related to genome editing. Tomorrow we will discuss multiplex editing, base editing, and other techniques. Okay, bye. And SDN1, SDN2 tomorrow we will discuss.
Sir,
Yeah.
Can I ask?
Yeah, you can ask.
Sir, the PAM sequence you told, that will be in target DNA, na, sir?
Yeah. PAM sequence is general, NGG. It is abundantly present in the genome.
Sir.
Yeah.
Oh,
Because
the
this
gene which is not having that PAM sequence, we will
no,
use
we can't design RNA for that. Okay. If there is no PAM sequence and this you should adjacent. It's not like that. It should include NGG, and you have to target the gene which are adjacent to this PAM sequence. If there is no PAM sequence, it is very difficult to design. No, you can't design. But it is regularly because it's just N is any base, just repetition of GG. NGG means what? N is any base, either A, T, GC, GG. You can occur many, like it will be the occurrence of frequency is very high. Every, like there is a frequency also. This occurrence of NGG, this also study is there, like how much frequency this NGG are any PAM sequence occurrence is very high. It is not that much a problem. Okay. But you are so unlucky that you don't have any PAM sequence that I had to think. I don't know that. Okay.
Oh, okay, sir. Fine.
Hello, sir.
Okay. Then ask.
Sir, in yesterday's class, you said that we can mutate a pollen and we can pollinate that pollen, sir.
Yeah.
After pollinating the seed we get, it is M1 seed, M1 generation.
No, I told transgenic, you self it, then you are getting M1.
Yes. That M1 is chimeric, sir, or is it fully mutant?
No, that can be possible, but which one you are? No, it is genomic. No, it is full mutation. See, chimeric, non-chimeric. While in first, when deciding mutation only, you have to decide that one. Either it is a chimeric or this one. While deciding mutation only, you have to decide. But after having a mutation, you definitely know because it's heritable means it should be there in the gene only. Some mutations will occur in somatic cells also, but they are not inherit to the next generation.
Yes, sir.
Okay.
If we treat pollen, sir, then it is only single cell, na, sir. Only single cell. The entire
No, cell you
cells will
remember.
get.
Nucleotide. Talking about nucleotide. Yeah, it may be chimeric. Like, see, you are mutating pollen. It will be see, whatever you do, it will be hemizygous in nature in M0. Sorry, M0. After allowing for selfing, it will become M1. Okay. Then you are getting segregation heterozygous, hemizygous, and normal. Okay, normal homozygous. This is the possibility. That chimeric, what is the meaning of chimeric?
The
The
plant body
presence of two or more different
different
type of
genotypes.
genotypes in a single cell.
Yeah, that is called hemizygous because it having two different things. One allele is having a gene, and another allele, sorry, another in a 2n condition, one is having gene and another having not gene, no gene. Because chimera can be defined in n number of ways. Chimera may be in terms of a cell, like in terms of a cell you take. Some part may be your other plant and some part may be other plant. And you take the chromosome level, some chromosome portion may having the other crop species and some of the portion from the other portion. This can be done. This is possibility is there. That is maybe hemizygous nature. That is hemizygous nature, no? Because it having both the genome, like one is, but it having the donor genome, but counterpart allele is not present. That is hemizygous. It's a chimeric nature only. But you have to fix it. You have to bring that into homozygous. That is the point. Understood?
And
Sir, if
sir,
we
another thing.
Yeah.
Yeah, please, Satya, please.
Yeah.
Sir, if we treat a seed, then the plant which are generated from those seeds, those are chimeric, sir, because seed is a multicellular. It may be multicellular in stage. The embryo might be in multicellular stage while we treating, and if we
Yeah.
germinate it, the plant may grow into a chimera. But if we treat pollen and we cross that, the plants may not be chimeric, na, sir. Definitely, the whole plant will be mutant only, na.
See, for example, if you are treating a seed, the mutation will happen. But we will write why the confusion. Treat a seed. Yeah. If it is mutation occur in a heritable portion only, the plant it will be M0. Yeah, definitely it will be hemizygous in nature. Then it is allowing it for pollen. The pollen which produce it's hemizygous in nature. Then you are crossing a selfing or whatever it may be with the pollen 2n. Then you are getting segregation for hemizygous, homozygous, and heterozygous. But you are treating a pollen having an allele, then crossing with normal. Yeah, we're getting a homogeneous. Definitely. You're correct. Actually, you are treating a pollen and you are using this pollen to cross with a normal individual. The progeny will be normal. Thank you. You are right. Okay.
It won't be a chimeric then. It is whole
Yeah.
mutant.
Yeah, but you are treating a seed, then definitely it will be that the pollen which produces the gametes which produced from this treated plant, it will be hemizygous in nature. But you are treating a pollen, then you are using this pollen to pollinate with a normal plant. Means you are going advancing, you are doing mutation here and you are using this one. Definitely it will not be chimeric. Exactly right. Okay. And I am very happy. Mm.
Why, sir?
No, really, because actually, no, it's correct only. I was forgotten. Actually, earlier I was not I was confused. I didn't think. I have not got your point earlier. Okay, while writing only it will be clear. Okay. Then any other notes?
Sir, without whole genome sequencing, we can do CRISPR, na, sir?
Yeah, we can't do. That's that because we need to design this one without information, without having information of sequence. It's very difficult to do. Okay. At least we should have a gene sequence. If you don't have whole genome sequence, at least you should have gene sequence. Then only we can able to design the RNA. Then we can do this one. At least gene sequences. If you identify the gene, like any other QTL mapping method, then you take that you can go for amplicon sequencing for particular region. Then you can design RNA for that. Then you can do that can be done. Even though you don't have whole genome sequence, you can do this. If you have, but the sequence of the DNA should must. Sequence of the gene is must. Without gene sequence, you can't do. First, we need to have map and we should have a sequence, then only we can do. But without whole genome sequence, that is a broader term because sometimes we know the some part like with gene sequence, but we may not know whole genome. That we can do. If you know the gene sequence, we can do. Even though we are not knowing whole genome sequence. Okay.
Okay.
But you can do now. Some part of the sequence you can take and you can sequence that. You can do. If you don't have whole genome sequence also. Okay, any doubts?
No, sir.
Tomorrow we will discuss some other part of the CRISPR-Cas9 like STN1, STN2. Sorry, STN and STN2 I will not cover because I have video. Please watch it. Like I made some animated video in YouTube. Uh, then after that we will discuss some multiplex editing methods and base editing, prime editing. We will discuss. It's a very interesting topic. This year question was asked base editing and what is prime editing. It's more advanced and what is CRISPR-12 and now I already we can edit RNA also because here we are editing the DNA by RNA, but we can edit RNA by RNA. Recently, I think one paper came. We will discuss that paper also, so that we will buy in genome editing. So we will discuss what are the different advantages of genome editing, what is multiplex editing, what is prime editing. We will discuss. Then we will wind up the class. Okay, bye. Good night, everyone.
Okay, sir.
Night.